Hello, students. Tonight, we will conclude our study of how the instructions in DNA are used to make the various proteins that give each of us our unique traits. Let's begin by looking at the figure to the right. This is a diagram of an unfolded protein. You can see that it is made up of smaller units called amino acids. So for instance, there, and there, and there, and so on. Each one of these bead-like structures is an amino acid, and strung together, they make the protein. In all, there are 20 different kinds of amino acids that the cells can use when building new proteins. Each different kind of protein that a cell makes is a chain of dozens or hundreds or even thousands of amino acids strung together in specific combinations. For example, the protein in our picture begins with 3 amino acids called alanine. There they are. Followed by an amino acid called methionine, and then lysine, and so on down the chain of amino acids. Now, in the same way that the order of letters in a word matters, the order of amino acids in a protein also matters quite a lot. Changing the order of the amino acids would actually change the protein that is made. Just like changing the order of the letters in a word, like this word here, for instance. Changing the order of the letters would change the actual word. So here, if I flip the first and the third letter, even though I'm using all the same letters, the order matters. These two words have different meanings and will change because of the simple change of the order of the letters. It actually changes the entire meaning of the word and potentially the entire meaning of the sentence. So, if we were to switch the order of the amino acids, say here, say we flipped those two and put them in different spots, or if we deleted some amino acids or added in extra amino acids, the type of protein, the identity of the protein that we would make would change altogether. This is important because if you change the identity of the protein, then you can't get done the original job that the original protein was intended for. So, it is very important that proteins get made with their amino acids in the correct order. Now recall, what is it that determines the order of the amino acids for a particular protein? What determines the specific order is the specific pattern of nitrogen bases in a gene. By reading the specific nitrogen bases in the gene, if the cell reads them correctly, and if those nitrogen bases are in the correct order to start with, then the correct protein should be made as a result of following the instructions in the gene. So, now we will describe the steps that are performed in a cell in order to make our very important proteins from the instructions in our gene. Okay, so this whole thing is how do we get a protein from the instructions in our genes. This process is very complex, but it is the key concept of this section. So I've simplified it into 3 parts for you. Pay attention carefully and please ask questions in class. All right, first, enzymes in the cell's nucleus will read the nitrogen base sequence of a gene and make a complementary copy of the sequence out of RNA. If we look at the picture here, I would like for you to imagine an enzyme attaching to the double helix. Let's say the enzyme attached right here. There's our enzyme. It's attached right there. And it's going to open this double helix, and then what it will do is it will read one side of the double helix. So this picture here is showing you with this arrow, it's showing you one side of the double helix with its base sequence magnified. As the enzyme reads the sequence A, C, C, A, C, C, G, A, T, G, T. As it's reading down that sequence, it is going to be making a copy of that sequence. But remember, the copy that is made is going to be a complementary copy, and it's going to use RNA nucleotides instead of DNA nucleotides. So, if what we just read off was the original strand, the enzyme, when it reads A, it's going to make a copy with the complementary base to A, which is U. And then it will move on, it will encounter the C, and then it will add the complement, which is G, to its RNA copy. And so on down the line. You can see that for each base the enzyme will read on the DNA, it is going to add the complementary base into the RNA copy. This process, where a DNA copy, the original DNA strand is copied into an RNA sequence, this process is called transcription. Okay. The purpose of making this RNA copy is so that we can move the information out of the nucleus. Okay, so this original DNA strand stays in the nucleus, and this RNA copy moves, and it's going to be moving out of the nucleus into the cytoplasm to find a ribosome. Okay. So the next thing that happens is the RNA sequence that moves out of the nucleus goes to a ribosome, and that ribosome will begin reading the RNA sequence 3 bases at a time, or in other words, in codons. So that's what is illustrated here. These 3 bases would be considered one codon, and the ribosome reads the whole strand in sets of 3s. So, each set of 3 bases is going to be a code for one kind of amino acid. For example, when the ribosome reads UGG, it knows to put in the amino acid tryptophan because the codon UGG is stands for the instruction to add a tryptophan to the amino acid chain. As the ribosome continues, it will read UUU. That codon stands for the specific amino acid phenylalanine, so it will add that amino acid next into the chain. And so on down the entire length of the RNA copy of the gene. As the ribosome reads the RNA sequence, it can use the code to string the amino acids together in the correct order. This process of using instructions in the RNA copy of the gene to make a correct functional protein, which is what we're ending with, is called translation. By performing the processes of transcription and translation, the cell is able to make all of the proteins it needs to grow, function, and give you all of your traits. Okay, we will continue to go over the processes of transcription and translation in class. I will show you several videos, and we will have several activities to review how this occurs. But we're going to move on to our last topic for tonight, which is the topic of genetic mutations. So, what happens if a mistake is made during DNA replication and the DNA in a particular gene is not copied exactly? For example, let's imagine new cells are going to be made, so the DNA shown in this diagram here needs to be copied so that we have the correct amount of new DNA for each cell. So, hopefully this diagram here looks familiar to you from when we talked about the process of DNA replication. I want you to focus in here, though, and I want you to imagine as the new DNA strand is being made, if the enzyme accidentally put a C, so this was a C, when it really should have put an A, like in this correct copy here. It's supposed to be an A, people. Okay, so if you put the wrong base in, now the instructions for making the protein that are contained within that gene are wrong. At least they're wrong in this DNA copy. They're still correct in this DNA copy. But the cell that gets this copy of the DNA will have the mutant copy, the incorrect sequence, and therefore the instructions for making the protein are incorrect. If the instructions are incorrect, then the protein that is made is going to be made incorrectly, and therefore the protein may not be able to do its job correctly. We call this type of occurrence a mutation, which we'll define here as any permanent change in a gene or in a chromosome of a cell. Now, the word mutation has all kinds of negative sounding connotations, but the reality is that while some mutations may be harmful, they may also be beneficial, or they may have no effect at all on the organism. Let's look at a couple of examples. In the first, the moth on the left here has a gene that has a normal color pigment, which gives it its normal brown color. The moth on the right, however, has a mutation. And the mutation is in the gene that codes for the protein that gives the moth brown color. Because of this moth's genetic mutation, it does not make the brown color appropriately, and therefore it results in a white color for the moth. In this case, however, the mutation in the right moth's color gene is actually beneficial to the moth because it can blend in with the light-colored bark of the tree and more easily avoid being eaten by predators. All right, now let's look at an example in which the mutation has a harmful effect. That's this one here. In the left side of the diagram here, you can see red blood cells from an individual that has genes to make the blood cell proteins correctly. In this side of the diagram, you can see red blood cells from an individual that has a mutation in the genes that make the blood cell proteins. Because this person does not make the blood cell protein correctly due to its mutation, his red blood cells will not be shaped correctly, which is why you see them shaped, flattened in these crescent shapes. Because they are shaped incorrectly, they will not be able to carry oxygen efficiently. This results in a disorder called sickle cell anemia, which can cause pain, weakness, and unfortunately an increased chance of an early death. Okay, so what besides incorrect copying can cause genetic mutations to occur in the first place? First, exposure to toxic chemicals like cigarette smoke, pollutants, and many other types can mutate your DNA. Additionally, overexposure to X-rays or UV rays can cause mutations to occur. So be careful in the sun and in any other situation in which you may be exposed to radiation. We will return to this concept of mutation many more times this year, as it is an important driving force in the evolution of species, as well as in the maintenance of our own health. For now, however, we are done. Don't forget your Quizmodo, and have a great night.